The main research that we're doing is we're looking for new cancer drugs from venoms. And I've got 2 PhD students working on this. One is looking at pancreatic cancer, colon cancer, and one is looking at breast cancer. And what we can do is we can grow these cells, we can grow human cells, we can grow animal cells in the lab, and we can treat them with different venoms and have a look at the effects to see if we can kill the cancer cells or slow their growth. And then we'll work out what it is in the venoms that's having an effect on the cancer cells, and then we can purify that, and then we can try and make that synthetically, so we can try and make it into a drug. There's a second part of our research, which is quite new and exciting, is we're looking for new antimicrobials. So at the moment there's a big problem with antibiotic resistance. So we're looking for, again with venoms, looking for components in the venoms that will actually kill bacteria. And we've got some very exciting preliminary results, but we've got a new master's student starting in September that's going to be working on this full time.
I'm interested in how cells communicate with each other. Essentially cells can emit like a signal. Essentially these are like cell bubbles, which act as like a biological email system. And we're interested to kind of decipher that signal, so what happens in, for example, in cancer or in Alzheimer's disease, as a way to detect for early stage of cancer. We've been currently working on different projects this year related to cancer, for example. In particular, we're looking at lung cancer, and we did a screen of 100 patients. Some were diagnosed with cancer, and we were able to detect for markers in the blood, which were found in these cell bubbles.
So my research project is focusing on developing a new way to validate and develop IVF media. IVF media is a form of nutrition that's provided to growing embryos in an IVF lab. So when the embryos are cultured in vitro, they're provided with nutrition through the use of media. This is usually validated with mouse embryos. So for my project, I'll be developing a new assay using cells from mouse embryos, so mouse stem cells. And I will use this assay to further develop IVF media produced by Genea.
So we are investigating prion diseases and protein folding diseases, and these are diseases that affect in mammals neurodegenerative diseases such as CJD in humans and BSC or mad cow disease, bovine spongiform encephalopathy in cows. And we are using a model organism, we're using yeast, good old baker's yeast, to investigate these phenomena, because yeast also get prions, and so we can use them. They're not contagious to humans, so we can study them safely in the lab.
We are looking to identify new anti-cancer drugs, and we're looking at animal venoms as a source for these drugs. So a lot of the therapies that we're currently using to treat cancer are starting to fail, so we need to look for drugs with new mechanisms.